So thank you very much, Catherine, for inviting me to speak. And I'd like to welcome everybody here to the Northwest Cancer Research Center. So I'm presenting, I guess, the viewpoint as the pathologist, and will try to explain to you over the next 25-30 minutes what happens to your intraocular biopsy. So who are the team in Liverpool? Well, there's Heinrich Heimann. There's also Karl Grönwald. They are the surgeons. And then they accompanying them in theatre are the operating assistants and also the porters who, in all their enthusiasm, support other charities as well. Here in November last year. So the background team, I guess, are the ones in the cellular pathology laboratory, which include myself, Luciane Irion, who is an eye pathologist in Manchester, who comes and reports here, Simon Biddulph and Anna Eichen, who are sitting here in the audience, who are the biomedical scientists who are dedicated for the eye pathology service. And then Dr. Helen Kellerai, Miss Sophie Thornton, who both provide the molecular prognostication and diagnostic service. So we've spoken about biopsies, and I guess, yes, you can already gather there are differing opinions across the United Kingdom, still united. And these are data from up to 2011, essentially, but there hasn't... There are increasing, so what you can see in this graph is, thank you, the number of new referrals, which are increasing, and I believe in 2014 was over 800. Of these new patients, there are melanomas about 250 to getting close to 300 in 2014, and about 60% of the patients have intraocular biopsies. So what are the biopsy indications? Well, there are a number of them. Small atypical melanocytic lesion, confirming the diagnosis. Choroidal melanoma, prognostication, which we'll go into in further detail. Differential diagnosis, is it a primary choroidal tumor or is it a metastasis? There are always these unusual lesions which can also occur in the choroids, the choroidal lymphomas. And then pigmentation following treatment, for example, plaque or proton beam. And then these indeterminate lesions, which even the best ocular oncologist in the world can't put 100% certainty make a diagnosis. So I'm essentially going to concentrate on the melanocytic lesions today. But we do, just to make clear, we do receive intraocular biopsies for other reasons and other tumors and conditions. So these are the main ones that we're going to be considering today. And you can see when you break down the data from the Liverpool Ocular Oncology Center that about 40% are for diagnostic reasons and 2/3 are for prognostication. So unfortunately, these videos don't work, so I've got to quickly go out of the presentation. These are the techniques which are applied. And this is the method of surgery, and I feel very inadequate speaking in front of surgeons describing. So this is a transretinal biopsy, going from the front of the eye through to the back of the eye. And essentially, this is without removing the vitreous. Go through the retina and do a biopsy of the tumorous area. And then this is then removed and sent to the pathology laboratory. Now there is the transscleral. So this is a tumor which is accessible through the sclera, and this is what's called a transscleral flap. First of all, the tumor is marked, and then a little flap of the sclera is created, and then a biopsy is taken, and then there is this superglue type used to seal the flap. And then finally, Amit mentioned that there were tumors at the front of the eye. And this is a biopsy of an unclear lesion. These are the so-called alligator or crocodile forceps, which were first produced in Essen. And there is a slight amount of hemorrhage or bleeding, which can occur, but you can see this sort of shark-like grabbing of the tissue, which is then viewed by the surgeon under the surgical microscope, and they swear to us pathologists that they always, it's like saying, look, Mama, I've caught a fish. It was originally this big, but actually it was this big. So this is the size of the tissue that we receive in the pathology laboratory. So just returning back to the PowerPoint. So these are the, I guess, the gung-ho surgeons who act on the crime scenes and provide us with all the evidence, you know, the clinical photographs, the biopsies. But the true people in the background who actually do the most important work are the cellular pathologists and the associated biomedical scientists and molecular technicians. So where are these people located? Yes, well, we still have the 4 centers where each of these centers have dedicated ocular pathologists and all the support staff. And so we have a specialized area of the large general pathology laboratory. So there are a large number of samples which come into, but we have a dedicated area. And since we are an ocular oncology referral center, we receive a large number of samples, not only from Liverpool, but from across the country and actually internationally. So we see a wide range of specimens. And I'll go into more detail, but so we see tiny biopsies consisting of only about 4 or 5 cells. And these would have been one of the clumps that you saw at the very end of that video, the iris biopsy. Then we have the local resections, which are larger samples, the enucleations, which Amit referred to, and then in worst case scenarios also the orbital exenterations. But I'm going to concentrate today on the intraocular biopsies. So after surgery, we get specimens in either these tubes or in these bags, cassette bags, and we also flush the tubing to make sure that we get everything out. And we sort of hold it up to light and compare it with what was described by the surgeons. Then Anna and Simon essentially take some of this fluid and they put it in a centrifuge. And they put it in a container. You can't really see it very well, but perhaps if I go to include this, they put some of the fluid into this container and then it's spun at a certain speed onto a slide. And then that is stained using conventional stains, which have been worked up for years in histopathology laboratories for then what's called morphological analysis. So we look at the cells. And so this is what we see under the microscope. So this is what's called a May-Grunewald-Giemser stain. And so this is a biopsy of a carcinoma or a cancer which is spread from elsewhere in the body to the choroid. And then these are typical examples of choroidal melanomas. And we can sort of tell on the basis of the cells, their particular characteristics, the color of the cytoplasm, what is melanoma and what is probably a cancer from another part of the body. But we then go on and do what's called immunohistochemistry, where we use particular markers which highlight it. And this is a stain against cytokeratin, which demonstrates that this is a carcinoma or a cancer arising from another place in the body. And we can then go on and do additional stains and work out whether these cancer cells are estrogen receptor positive, progesterone receptor positive, HER2 positive, etc., etc. And on the basis of this, then we can come up with a good estimate as to where the primary tumor is located. We can also do DNA extraction from these samples and do DNA mutational testing, which is important for choosing the chemotherapy for these patients. So this is in the case of metastases. And this is an example of an estrogen receptor on a breast carcinoma metastasis to the eye. But when we come to the choroidal melanoma biopsies, we essentially look down the microscope, and as I said before, they can vary considerably in terms of the degree of cellularity and also blood, associated blood with the biopsy. So here you have 3 or 4 tumor cells, but quite a bloody background in terms of a large number of erythrocytes. And then here you have a large number of tumor cells. So we assess for the overall degree of cellularity, the percentage of tumor cells within the biopsy, the degree of pigmentation, how heavily pigmented the cells are, as in how much melanin they have, cell viability and hemorrhage, and then we also extract the DNA. So we extract the DNA from the tumor cells. And we do this on not only pre-plaque biopsies, but also post-proton beam therapy. And you can see that the cells here are quite viable, despite the proton beam, and these biopsies are taken on the very last day. So on the basis of this, on the basis of the DNA concentration, then we do the genetic testing, and I'll go into a bit more detail about that. And then there are 2 main techniques which we use. One called MLPA for short, and I'll come back to that, and another called microsatellite analysis. And this is all on the basis of the concentration of the DNA in the sample. So the microsatellite analysis essentially looks predominantly at chromosome 3, which is the main chromosome with respect to prognosis in uveal melanoma. And we look at, we can say, 2 parts of chromosome 3, and we look at the biopsy, the tumor cells, and then we also look at the blood. We need the blood sample for comparison. And here there is no change in these 2 components in the tumor cells compared to the blood. So therefore, this is disomy 3 tumor. Okay. Here, in contrast, you have an alteration in this second part compared to the blood, and so there is a loss of chromosome 3. So this is monosomy 3. So this essentially, with microsatellite analysis, it's used for samples which have a small concentration of DNA. And it's looking at really only sort of 2 points on chromosome 3. MLPA, in contrast, looks at a wide range of different points along chromosome 3 and also other chromosomes 1, 6, and 8. And we can use this technique for fresh samples and also for fixed samples. So we receive blocks from other pathologists across the country and indeed the world to perform this test. So we extract the DNA, we do the quality control, we do the MLPA, as in Helen and Sophie do, they do the interpretation and the data analysis and then bring it all together. So this is a typical musical score, we call them. So these are the control probes. This is chromosome 1, this is chromosome 3, where there is loss on several points along chromosome 3. Here's chromosome 6, chromosome 8. So this is a monosomy 3 tumor, and here there are no alterations on chromosome 3, and so that is a disomy 3 tumor. And we have processed these quite rapidly. You can imagine that this whole process does take time, but our mean turnaround time is 7 days. So in the meantime, we've done approximately 700 cases. This is data to 600 cases, UK-based patients, and we can quite confidently divide the tumor types into those with a high and a low risk of spreading, and perhaps the worst combination to have is alterations on chromosome 3 and amplifications on chromosome 8. And we've then gone on and analysed this together with a biostatistician and doing so-called cluster analysis. And we can quite clearly divide, using chromosome 3, the tumours into 2 groups. And you can see this again here. There are 2 quite clear groups of tumours. And then when you combine the chromosome 3 and 8, you still get that, but there are some tumors which have some overlap. And these are the so-called gray zone cases. And so there, here you can see there are two distinct groups. However, there are some tumors where we really aren't sure about these cases, which direction they're going to go. And whatever method you use, whether it's gene expression profiling, or what we're using, or more complicated techniques, we're still going to get these gray zone cases, unfortunately, and we're trying to work on this to help define the prognosis better. And this map is, this diagram essentially was the gene expression profiling, which came up with the so-called class 1 and class 2 terminology. Now there is subdivision of these tumors into class 1A and class 1B. Nobody really knows what 1B means. And with respect to comparison of the different techniques, there's a lot of controversy. The published data are flawed. There are various weaknesses to the different studies, retrospective, the small number of cases, etc., etc. So there is no gold standard, despite what you may hear from particular companies. There's no gold standard with respect to which technique should be used. We did do an analysis comparison of MLPA versus gene expression profiling and found that there was very good consensus between the two techniques. However, this does cause quite a bit of heat generated in some discussions, and not only in conferences but also in the literature. But I think the main point to emphasize here is that these techniques, they're not 100%, but we're working on it to improve their accuracy. But the strongest recommendation is that you, like any molecular pathology tests, they're not standalone. You do incorporate them with the clinical and the histomorphological findings. And this is what essentially is done in Liverpool, is use this prognostication model, the so-called Liverpool Uveal melanoma prognostic indicator. And this takes into account a whole range of different factors. And for each single individual patient, a prognostic curve is generated, and then the patient is counseled with the ocular oncology team accordingly. And all tumors are also TNM staged. So tumor biopsy and genetics, yes, it is controversial, but if you were to look at the fundus and say, what is the prognosis of this patient? Without biopsy, then you are relying only on the clinical features and with respect to location and size and age and gender. You add the genetic data and the curves change, as you can see, at the monosomy 3, and there is these curves are unfortunately show a worse prognosis. This data is based on this prognostication model is based on a very large database, and there was a test and then validation set produced by the biostatisticians, and it is quite robust. So it is controversial. People say, as you've heard, that a diagnosis is actually achievable without biopsy. Well, there are cases which still you'd have 10 ocular oncologists around the table and they would not agree whether it is a melanoma or not. Most patients don't want it. Well, that's also debatable. I guess it depends how the information is given to the patient and the degree of consultation. No treatment for metastatic disease. I think that is going to be soon an outdated statement. And I'm not going to steal Joe's thunder, but by identifying high-risk patients, then they can be screened more intensely and using MRI plus or minus contrast, pick up the metastases earlier with the view of then removing them surgically. And certainly data from Liverpool and also from Paris would indicate that the survival of these patients is lengthened. And there's very preliminary data that a MEK inhibitor does result in prolonged progression-free survival. And there are numerous targets on the horizon. We're better understanding what's going on in uveal melanoma cells, not only in the primary tumors but also in the metastases, and that it has become a bit of a trendy tumor to research into. 5 years ago, you wouldn't have found any papers on uveal melanoma in the very high-ranking cancer journals. Now you see them in Nature and Cell and Science. So it is a trendy area, and the drug companies are actually wanting to become involved. And very recently, this new pathway was discovered where they're activating mutations, which is attractive with respect to coming up with new therapies. So there were the suggestion that there also was a complication rate, and I'm going to probably leave the discussion for that to Heinrich, but this is a paper which was recently published in Ophthalmology, and Martina Andy, who's at the back, was involved in this. But you can see vitreous hemorrhage tends to be transient, but if it is persistent, then can be removed with a vitrectomy. Similarly, retinal detachments are rare, but can be treated, and endophthalmitis, which is a risk with any intraocular surgery, is exceptionally rare in this cohort. Seeding, yes, also a problem. There were 3 cases here in Liverpool, but they have since been controlled with additional plaque therapy, and all patients are alive without any evidence of metastasis. So, and then there is despite all efforts from everybody, the disappointed patient who, for some reason or another, we weren't able to come up with a genetic result. And I'm not going to steal Laura's thunder, but this is obviously one of the major concerns post-op of the patients. We've done an audit, which we haven't published as yet, but we're getting there, where we've subdivided the biopsies into the sort of our initial learning phase and from 2012 to 2014. And yes, at the beginning, we found that we had a poorer rate in coming up with the genetic testing results, particularly in small tumors. But since then, we've improved on a variety of different things: the surgical technique, the delivery of the sample, the working up in the lab, et cetera, and we really squeeze as much information as we can out of these biopsies. And it's very rare on the basis of cytomorphology that we can't make a diagnosis. And certainly with the genetic testing, we're decreasing that from 24 to 8%. So we're trying with each year and technique, we're trying to improve the yield from these biopsies. So what do we do in the daily practice? Well, if it's an indeterminate lesion, there are 3 options which are always discussed. Observation, which was discussed before, biopsy, and or just treat. Choroidal melanoma. If it's clear it's a choroidal melanoma, then always discuss the prognostic biopsy. Always discuss the risk that it could be non-productive, the potential collaborations, including the seeding and any other ones. It is very much a joint decision, and patient's choice is included, and there is a very good psychological support team in the Ocular Oncology Centre here, and also a budding system which has been set up locally and I think also nationwide. So there's more information regarding the intraocular biopsies and any sorts of samples that you'd want to send to the pathology laboratory, not only for patients, but clinicians and also for researchers. These are some of the team at work. And I think that might be it. Well, thank you very much for your attention, and I'm willing to take any questions.
Questions from the floor?
Hi.
Is it possible to do a prognostic biopsy after treatment?
So is it possible to do a prognostic biopsy after treatment? I guess it depends on what type of treatment you've had. Yes, so there was one slide which I showed you. On the very last day of proton beam therapy, an intraocular biopsy is taken, and we work that up as we would any other biopsy, for example, a patient who's been treated with plaque. Depends how long you've had your somebody's had the proton beam therapy, we haven't evaluated whether you could get a result from a patient who's, say, been treated a year ago and has persistent non-active tumor in the eye, as to whether we would be able to get a result of that. And I don't think anybody's done that as yet.
Just a question on the reference there of the chromosome 3. Is it such now that the research, you're delving into the genetics of the etiology of this disease, that you've identified the faulty alleles within these 2 or 3 chromosomes that you made reference to?
No, it's so the question was whether we have identified the faulty genes which have caused the tumor on chromosome 3. Unfortunately, no, that's not what we've identified are parts of chromosome 3 which are often altered and in the tumor cells. And so, so we target those particular parts of chromosome 3 to see whether there are changes in what's called copy number. And if you have the normal copy number, then in all of these points along chromosome 3 and 6 and 8, then we can quite confidently say this is disomy 3 or disomy 8, etc. But there is increasing evidence that there are causal mutations resulting in the development of the disease. And one of those is BAP1, which I didn't mention here. And BAP1 is a gene which is found on chromosome 3, but it's found in all cells. So this is what's called a germline mutation. So it's a bit complicated. So what we analyze in the tumor cells are called somatic mutations. But if you are born and you have underlying so-called germline mutations in every single cell of your body, and should that be a BAP1, a mutation, then you have an increased risk of developing not only uveal melanoma, but potentially other tumors. And so there have been, in the recent literature, descriptions of some families who've got these so-called germline BAP1 mutations. And these families have an increased incidence of uveal melanoma, some lung tumors, some kidney tumors as well. And so what we're planning on doing, and we have a fellow who's analysed all of our data, and he's pulled out patients who've had more than one tumour. So they've had a uveal melanoma plus another tumour. And what we'd like to do is go on from that and take blood samples from these patients, if they're agreeable, to see whether they have an underlying germline mutation, which essentially makes their whole genome unstable, and so that they can have this increased risk of developing a number of different tumours.
Unfortunately, we don't know what causes the melanoma. It suggests something on chromosome 3 must be the reason, obviously, but we still have patients that have monosomy 3 and never get metastasis, and we have patients with metastasis that have a normal disomy 3 test. So there are things we don't understand, but just if you take all the risk factors that we can look at, this is with the highest correlation with metastatic disease, yes or no, but we don't know why.
Can I ask a question about that? About biopsies, about what you just said about the disomy 3, people sometimes getting it. Is it possible to do like a biopsy and to get 2 different results if you took it from different parts of the tumour?
That's a good question. So that's, yeah. Repeat the question. So is it possible to have 2 different results from differing parts of the tumour? Yes, it is. And this is called tumour heterogeneity. It is, however, quite rare. So we've done a concordance study looking at intraocular biopsies, and then for some reason or another, the patients have had a larger operation, either an endoresection, a local resection, or a nucleation. And then we've compared the results of that larger specimen with the intraocular biopsy, and we've had 100% concordance with results. However, there are always exceptions to the rules, and there was a case that we also published where at the base of the tumour, it was quite benign looking from the cell type. There were spindle cells. We had a disomy 3 result using the MLPA. But at the top of the tumour, very aggressive looking cells, epithelioid, high mitotic rate, and we did the MLPA from there, and there was a monosomy 3. So there are always these cases, and so there are always exceptions to rule, but I think the surgeons do try and go and do 2 or 3 passes. And so we analyze the tumour, always looking at the cell type. I think it is, there are some places where no morphological assessment is carried out, so you actually don't know what cells you're analyzing, which is a problem if you're actually not analyzing tumour cells, as in melanoma cells. And that has been reported that you get a disomy 3 result on perhaps a tumour which was not a melanoma, but was a carcinoma, and which normally doesn't have any alterations on chromosome 3 anyway. So this is why we are a very strong belief that you have to have morphological analysis. Yes.
Is it possible that a sample for biopsy of a tumour would be different before proton beam therapy to after proton beam therapy?
So the question was whether there's a possibility that the result of an intraocular biopsy before and after proton beam could be different. We haven't published this yet either. This is something that we have to get on and do, but we do have such comparisons as well. And the results for the MLPA are the same. And I've heard from Paris, the group in Paris who do the genetic analysis using a different technique called array CGH, that they also have done this comparative study, and there is no difference in the results. It depends on the timing of the biopsy after the proton beam. And so it has to, well, the evidence so far would suggest that it has to be performed quite soon after the end of the proton beam therapy.
Correlating with metastasis, the size of the tumor, okay? And you always have to look obviously at the whole patient and the story behind it. So the smaller the tumor, the better your chances of not having that, and the higher the risk of the biopsy. So the second thing is, where is the tumor? If the tumor is in the periphery, it's easier, and usually your risk will be getting a hemorrhage or retinal attachment, which in the worst of cases usually can be treated. Whereas a very small tumor at the posterior pole, you run a risk of causing problems to your vision as well. And there comes sort of your risk-benefit ratio changes then. So let's say you have a small tumor at the back of your eye, close to your center of vision. Your risk of this being metastatic is 10% or 15% or so. The risk of the biopsy of getting a problem is 5%, 10%. And not getting a result, and the worst thing that could do is induce a visual problem and not get a result afterwards. Then you'd be very disappointed as a patient and a surgeon as well. So we discuss it that way, obviously, and it really entirely depends on the patient. And I mean, Laura is going to talk about this. It's a very complex issue, and there are lots of factors play into it. You know, in one way you want to comply with what's offered. You think also it may be good for research purpose, but there may be something in the back of your mind you say, I don't want this really. And this is why we usually try not to pin down that decision during the first discussion, but always give some time to reflect on it, talk to somebody else, and then come to a decision. What we don't want is that you make the decision to please anybody, or because you think, oh, that's how you do it or so. It is a very complex issue, and Laura talked to hundreds of patients, and I think she will do that better. But there are some patients, and the basic rule is the more posterior, the smaller, the higher the risk, and the lesser the risk of it being high risk. So, but we do have patients, even in mean, very small lesions who absolutely want to know, and why should I say no? We offer it to anybody. Another question, please.
I'm a patient who had a biopsy, and I find the information very helpful that I was at high risk. My question relates to the elimination of controversy. My biopsy was analysed using a fish technique. I don't really understand what that is. But has that now been superseded? Or is this where the controversy is?
So, yes, the question is, so the gentleman has had an intraocular biopsy and it was evaluated using fish. And on the basis of this, you were given information that it was high risk. And is it controversial regarding surgical complications, but is adding to the controversy, is it the method used? Definitely, whether fish has been superseded. So we, Professor DeMar, so fish is fluorescence in situ hybridization. Is it just a fancy technique, which essentially you have particular probes which target parts of particular chromosomes. And so fish was introduced into Liverpool as the main technique for prognostication, and that was in 1999. Then in 2007, we did an audit analysing all of the results of the fish. And at the beginning, the fish probes were only for a particular point of chromosome 3. And it was actually at the middle. And chromosomes have 2 arms at the top and 2 arms at the bottom. And so if you've got a probe which is only at the middle of the chromosome, then you don't pick up the minor alterations which could go on in the 4 arms. And so we found that in this audit that there were results going out which were inaccurate. So saying that the tumour cells were disomy 3, but unfortunately the patients did indeed develop metastases. So people then started adding probes, so one of the arms and one to the others. But this it requires more material and is a bit fiddly. So we then moved on to MLPA. We did an initial research step, validated the method, and then brought it into the clinic as in routine diagnostic work. And it analyzes initially 13, now up to 18 different spots on all arms of chromosome 3. And so it increases the accuracy of you picking up any alterations all along. And it's not only looking at chromosome 3, but it's looking at chromosomes 1, 6, and 8, which are also considered to be important. So our feeling is that fish is not enough, that you really need to do something with a higher resolution, a technique which is also affordable by the NHS. So, for example, gene expression profiling is hugely expensive and wouldn't be affordable for every single patient in the NHS.